Welcome to the Deep Sleep Science channel. I'm so glad you've joined me tonight. Tonight's journey takes us into the smallest corners of existence, a world that unfolds beneath the threshold of sight. All around you, invisible to the naked eye, an entire universe pulses with motion. Water molecules dance, atoms spin in their silent orbits. Microbes drift through microscopic seas. This hidden realm has been here all along, woven into every breath, every drop of water, every grain of soil. The microscopic world is often overlooked, sometimes feared, but it is truly one of the most beautiful and intricate dimensions of nature. Within a single drop of pond water lives a cosmos of creatures moving with purpose and grace. On your skin, in your gut, in the soil beneath sleeping trees. Trillions of microscopic beings carry out their quiet work. Science has only begun to map this vast and infinitesimal frontier, and what we've discovered so far is nothing short of miraculous. Tonight, we'll drift through 100 gentle facts about this invisible world, where the smallest things reveal the deepest wonders. If you enjoy these gentle journeys, consider liking and subscribing, or share your thoughts in the comments. It helps others find their way here too. But for now, there's nothing you need to do. Just let yourself settle, let your breath slow, and let the hidden universe of the microscopic world open like a calm horizon beneath your closed eyes. Now, if you're ready, let's begin. A single drop of seawater contains millions of microscopic organisms. If you were to hold an eyedropper above the ocean and capture just one glistening bead of salt water, you would be holding a universe in miniature. Within that drop, millions upon millions of tiny beings live out their entire lives, drifting with currents too subtle for us to feel, feeding on nutrients too small for us to see. There are bacteria that glow with bioluminescent light, plankton that spin like living wheels, and larvae of creatures that will one day grow large enough to meet our gaze. Each organism moves with ancient purpose through its microscopic sea, unaware that it shares its world with countless others, unaware that the ocean itself is nothing more than an infinite collection of such drops. It is a reminder that abundance exists at every scale, that life finds a way to flourish even in the smallest of spaces. Tardigrades, also called water bears, can survive in the vacuum of outer space. These microscopic creatures, no larger than a grain of salt, possess a resilience that defies our understanding of what life can endure. When scientists expose tardigrades to the void of space, where there is no air, no warmth, no protection from cosmic radiation, the tiny animals simply entered a state of suspended animation and waited. They dried out completely, their bodies becoming almost crystalline, and yet somewhere deep within their microscopic cells, the spark of life remained. When returned to water and gentler conditions, the tardigrades awakened, resumed their slow bear-like walk, and continued living as if nothing extraordinary had happened. They have survived temperatures near absolute zero and heat that would boil water. They have endured pressures 6 times greater than those at the bottom of the ocean. Their survival is not a miracle, but a patient adaptation refined over millions of years. In their tiny resilience, we glimpse the stubborn, quiet determination of life itself, waiting through impossible conditions for the chance to continue. Diatoms are microscopic algae that look like fragments of stained glass. If you could shrink yourself down and swim through a drop of pond water, you would find yourself surrounded by living jewels. Diatoms are single-celled organisms encased in silica shells, and each shell is a masterpiece of geometric precision. Some are shaped like pillboxes, others like stars, still others like intricate snowflakes made of glass. Under a microscope, they shimmer with patterns of holes and ridges, each species crafting its shell according to an ancient blueprint encoded in its genes. These microscopic artists drift through oceans and lakes by the trillions, capturing sunlight and turning it into oxygen, their glassy bodies refracting light in ways that make them appear to glow. When they die, their silica shells sink slowly to the ocean floor, layer upon layer, forming deposits that have been pressed into stone over millions of years. The chalk cliffs of Dover, the diatomaceous earth we use in filters and gardens, all of it was once a silent rain of these glass-encased cells, falling through dark water. They remind us that beauty exists at every level, that even the smallest builder leaves a mark upon the world. Water molecules are constantly moving, even when the water appears perfectly still. When you look at a glass of water sitting undisturbed on a table, it seems the very image of stillness. But if you could see through to the molecular level, you would witness an endless storm of motion. Each water molecule vibrates and spins, colliding with its neighbors millions of times per second. The molecules at the surface leap into the air as vapor, while others fall back down to take their place. The liquid shifts and flows internally, currents too small to measure swirling within the glass. This motion never stops, not even when water freezes into ice, for the molecules still vibrate within their crystalline lattice, trembling with the energy that defines all matter. The stillness we perceive is an illusion created by the limits of our senses, a calm surface hiding an invisible dance beneath. It is a profound reminder that peace and motion are not opposites, that even in rest there is eternal movement, and that the universe at its smallest scale never truly sleeps. Your body contains more bacterial cells than human cells. You are not a singular organism, but a walking ecosystem. For every cell that carries your unique human DNA, there are slightly more bacterial cells living within you and upon you, going about their microscopic lives. They cluster in the folds of your intestines, on the surface of your skin, in the warm cave of your mouth. These bacteria are not invaders, but partners, ancient allies that arrived even before you were born and have shaped who you are in ways we are only beginning to understand. They help you digest food that your own cells cannot break down. They produce vitamins that keep you healthy. They train your immune system to recognize friend from foe. Some scientists believe that what we call you is actually a collaborative community, a symphony of human and microbial life working together toward a common goal. The bacteria within you have been refined over millions of years of evolution, and their presence is not a contamination, but a completion. You are never alone, not in the truest sense, for you carry within you a hidden world of helpers too small to see, but impossible to live without. Atoms spin silently at the foundation of all matter. Every object you have ever touched, every surface you have ever seen, every particle of your own body is made of atoms, and at the heart of each atom is a kind of motion so fundamental that it has never stopped since the beginning of time. Electrons orbit the nucleus in clouds of probability, not like planets in fixed paths, but like whispers of presence, existing in multiple places at once until observed. The nucleus itself, made of protons and neutrons, vibrates with an energy that has persisted for billions of years. These particles do not rest. They spin with an intrinsic property called angular momentum, a rotation that is built into the very fabric of what they are. If you could zoom in far enough, you would find that the solid table beneath your hand is mostly empty space, filled with these spinning, dancing particles, held together by forces we cannot see. The stillness of the world is an illusion woven from invisible motion, and the silence we perceive is actually a constant hum of energy, too small and too fast for our senses to detect. In the spinning of atoms, we find the heartbeat of existence itself. DNA molecules can jump from one location to another within a cell. Inside the nucleus of nearly every cell in your body lies a tightly coiled library of instructions, the double helix of DNA that defines who you are. But this library is not static. Hidden within your genetic code are sequences called transposons, sometimes known as jumping genes, and they have the remarkable ability to move themselves from one place to another along the strand of DNA. They copy themselves and insert their copies elsewhere, rewriting the genetic text in subtle ways. These jumping genes were once thought to be useless relics, junk left over from evolutionary history, but we now know they play crucial roles in genetic diversity and even in the way organisms adapt to changing environments. In a sense, your DNA is not a fixed blueprint, but a living document, constantly being edited at the microscopic level by these restless wanderers. The jumps happen without sound, without fanfare, in the quiet darkness of the cell's nucleus, yet they can change the course of life itself. It is a reminder that even the deepest instructions of biology are not written in stone, but are fluid, adaptable, endlessly creative. Scientists estimate there are about 1 trillion different species of microbes on Earth. When we think of biodiversity, we often imagine rainforests filled with birds and insects, coral reefs teeming with fish, savannas where great herds roam. But the true abundance of life exists far below the threshold of our vision. In the soil, in the oceans, in the air, in the deepest rocks beneath the surface, there are microbes of staggering variety, each species uniquely adapted to its microscopic niche. A single handful of soil can contain more species of bacteria than there are species of mammals on the entire planet. A teaspoon of seawater holds communities more diverse than any city. These microbes have evolved across billions of years, radiating into every conceivable environment, from boiling acidic hot springs to frozen Antarctic lakes locked beneath miles of ice. They are the unseen majority, the quiet architects of ecosystems, the foundation upon which all visible life rests. And yet, we have only just begun to meet them. Most remain nameless, their existence inferred but not yet confirmed, waiting in the invisible world to be discovered. We've discovered less than 0.001% of all microbial species. For all our scientific progress, for all our microscopes and genetic sequencers and advanced technologies, we have only scratched the surface of the microbial world. Of the trillion species scientists estimate exist, we have formally identified and studied fewer than 10 million, and even that number may be generous. The vast majority of microbes cannot be grown in laboratories, and so they remain mysteries, known only by traces of their DNA floating in samples of water, soil, and air. Each newly discovered species reveals unexpected capabilities, new biochemical pathways, novel ways of surviving in conditions we thought were impossible for life. There are microbes that breathe metals instead of oxygen, that feed on radiation, that thrive in darkness kilometers beneath the Earth's crust. We are explorers standing at the edge of an invisible continent, and every step forward reveals how much further the horizon extends. The humility of this unknowing is itself a kind of comfort, a reminder that the universe still holds endless secrets, and that wonder is always waiting just beyond the limits of what we can see. Tardigrades are only 0.1 to 1.5 millimeters long, about the size of a grain of salt. If you were to place a tardigrade beside a grain of table salt, they would be nearly the same size, and yet the tardigrade is infinitely more complex. Within that tiny body, barely visible to the naked eye, there is a complete organism with a brain, a digestive system, 8 legs tipped with claws, and the ability to survive conditions that would obliterate almost any other form of life. It breathes, it eats, it moves with deliberate purpose across the wet film of moss or lichen where it makes its home. Under a microscope, its stubby legs churn in a slow walking motion that is strangely endearing, almost comical, as if a bear had been shrunk down to the size of a speck and set wandering through a forest of moss. Despite their microscopic scale, tardigrades have been found on mountaintops and in ocean trenches, in tropical rainforests and Arctic tundra, thriving quietly wherever there is a thin layer of water to sustain them. Their smallness is not a limitation, but a kind of freedom, for they slip between the cracks of the world and persist where larger creatures cannot. The largest bacteria ever discovered can be seen without a microscope. In the mangrove swamps of the Caribbean, scientists found a bacterium that defies every assumption we held about the microscopic world. This organism, named Thiomargarita magnifica, can grow up to 2 centimeters long, roughly the size of a human eyelash, large enough to be seen with the naked eye and picked up with tweezers. For a bacterium, this is incomprehensibly vast, thousands of times larger than most of its relatives. It exists as a single cell, yet within that cell is a complexity that borders on the architectural. Long filaments stretch through its interior like support beams, and its genetic material is carefully organized in ways that more closely resemble the cells of complex organisms than the simple bacteria we thought we understood. The discovery of Thiomargarita rewrote the boundaries of what we thought was possible for single-celled life. It reminds us that the categories we create, the neat divisions between microscopic and visible, between simple and complex, are often blurred by nature itself, which refuses to be constrained by our definitions. Some giant bacteria look like strings of pearls under magnification. Among the larger bacteria that challenge our definitions, there are species that arrange themselves into delicate chains, each individual cell glowing like a luminous bead threaded onto an invisible string. These bacteria live in the sulfur-rich sediments of the ocean floor, and they survive by processing chemicals that would be toxic to most other forms of life. Under a microscope, they are hauntingly beautiful, their translucent bodies lined up in perfect rows, shimmering with the granules of sulfur they store inside themselves. The chains can stretch for centimeters, visible to the careful eye, and they sway gently in the currents of their aquatic world. They are ancient organisms, descendants of some of the earliest forms of life on Earth, yet they continue to thrive in the quiet darkness of deep water, processing elements, building energy, existing in a state of patient stillness. They are living proof that even the simplest forms of life can create beauty, that elegance is not reserved for the complex, but is woven into the very foundations of biology. Listeria bacteria move through cells with glowing comet-like tails. When certain bacteria invade a living cell, they do not simply float passively within their cellular prison. Instead, they hijack the cell's own machinery to propel themselves forward. Listeria monocytogenes, a bacterium that can cause food poisoning, has evolved a remarkable strategy for movement. It triggers the cell to assemble long filaments of a protein called actin behind it, and these filaments push the bacterium forward like a rocket trail. Under fluorescent microscopy, where the actin filaments are stained to glow, the bacteria appear as tiny comets streaking through the cellular landscape, each one trailing a brilliant tail of light. They use this motion to thrust themselves through cell membranes and into neighboring cells, spreading through tissue without ever exposing themselves to the immune system patrolling outside. It is a form of travel both elegant and sinister, a microscopic ballet of invasion and adaptation. Watching these glowing trails, it becomes impossible not to admire the ingenuity of life, even in its most unwelcome forms, for evolution has equipped even the smallest organisms with strategies of breathtaking sophistication. Tardigrades can survive without food or water for years. If conditions become unfavorable, if the film of water where a tardigrade lives begins to dry, the tiny creature does not panic or flee. Instead, it performs a transformation that seems closer to magic than biology. It retracts its 8 legs, expels nearly all the water from its body, and enters a state called cryptobiosis, a kind of living death where metabolism slows to an undetectable crawl. In this desiccated form, called a ton, the tardigrade can endure for years, even decades, waiting with infinite patience for water to return. During this time, it does not eat, does not breathe, does not grow. It simply exists, suspended in a liminal space between life and non-life. Its biological processes paused, but not extinguished. When moisture finally arrives, the tardigrade absorbs the water, unfurls its legs, and resumes its slow walk through the moss as if no time had passed at all. This ability to pause life itself, to wait through impossible droughts, and then awaken unchanged, speaks to a resilience that transcends our usual understanding of survival, a testament to the stubborn endurance embedded in even the smallest threads of existence. The microscopic world exists on your eyelashes right now. At this very moment, as you lie still and listen, there are tiny inhabitants living among the roots of your eyelashes. They are called Demodex mites, microscopic arthropods with elongated segmented bodies and stubby legs, and they make their homes in the follicles where your lashes grow. They are so small that dozens could line up end to end across a single eyelash, and they spend their entire lives in this warm, sheltered landscape, feeding on oils and dead skin cells. Most people carry these mites without ever knowing it, for they cause no harm and go about their quiet business in the darkness of closed eyes and the gentle rhythm of blinking. They are born, they live, they reproduce, and they die, all within the tiny universe of your face, never traveling more than a few millimeters from where they began. It is a strange and humbling thought that you are not just a body, but a habitat, a living terrain that supports invisible communities who know nothing of the larger world beyond. In their small lives, we are reminded that perspective changes everything, that what seems vast to them is imperceptible to us, and that every scale of existence contains its own quiet dramas. A single human cheek cell is invisible to the naked eye, but contains a universe of molecular machinery. If you were to gently scrape the inside of your cheek and place the cells on a microscope slide, you would see rounded shapes, each one a self-contained world. The cell membrane holds everything together like the walls of a city, while inside, the cytoplasm flows like a living river carrying nutrients and waste. The nucleus sits at the center, a library containing the entire instruction manual for building a human being, written in the 4-letter alphabet of DNA. Mitochondria drift through the cell like power plants, converting sugar and oxygen into usable energy. Ribosomes assemble proteins letter by letter, following the instructions delivered by messenger molecules. The endoplasmic reticulum folds and packages these proteins, preparing them for export. The Golgi apparatus tags and ships them to their destinations. All of this happens simultaneously, a coordinated symphony of molecular interactions so complex that we are still discovering new components and pathways. A single cell, too small to see without aid, contains more organized activity than the busiest city on Earth. And it performs this work flawlessly, billions of times over, every second of every day, maintaining the miracle of your existence without conscious thought or effort. The human eye can only see objects larger than 100 micrometers. There is a barrier built into the very structure of our vision, a threshold below which the world becomes invisible. The resolution of the human eye is limited by the physics of light itself, by the wavelength of the photons that enter our pupils, and the spacing of the cells in our retinas. Objects smaller than about 100 micrometers, 1/10 of a millimeter, blur into nothingness, their details lost to us no matter how closely we look. This means that entire categories of life and structure exist just beyond the edge of our perception, present but unseen, real but inaccessible. The intricate geometry of pollen grains, the graceful swimming of paramecia, the crystalline architecture of diatoms, all of it unfolds in a realm we cannot enter without tools. For most of human history, this microscopic world was not merely unseen but unimaginable, a dimension of reality that did not exist in human consciousness. It was only with the invention of the microscope that this veil was lifted, and we discovered that sight is not truth, that the boundaries of our senses are not the boundaries of existence, and that the universe extends infinitely in both directions, toward the vast and toward the infinitesimal. A single strand of human hair is approximately 100 micrometers wide. If you were to pluck a hair from your head and examine it closely, you would be holding something that exists right at the threshold of microscopic scale. It is just barely visible, thick enough to see and feel, yet thin enough that its internal structure remains hidden from the naked eye. Under a microscope, a hair reveals itself as a complex, layered material. The outer cuticle is made of overlapping scales, like roof tiles, that protect the inner cortex where pigment and structural proteins reside. The very center may contain a hollow medulla, a channel that runs the length of the shaft. These layers work together to give hair its strength and flexibility, its ability to bend without breaking, and to grow continuously from the follicle. The width of a hair has become a standard unit of comparison in microscopy, a reference point for describing things at the edge of visibility. When we say something is thinner than a hair, we are invoking this boundary, this place where the visible world gives way to the invisible, where the familiar dissolves into mystery. Microscopes allow us to see objects as small as 0.1 nanometers. The invention of the microscope was one of humanity's great leaps into the unknown. A doorway opened into a dimension that had always existed but never been witnessed. With glass lenses and focused light, we pushed back the boundary of visibility and discovered entire kingdoms of life that had been invisible to every generation before us. As technology advanced, our microscopes grew more powerful. Light microscopes gave way to electron microscopes, which use beams of electrons instead of photons to illuminate the infinitesimal. These instruments can resolve details as small as 1/10 of a nanometer, a scale so tiny that we are looking at the spaces between individual atoms. At this magnification, the familiar world dissolves entirely. Cells become landscapes. Molecules become visible as distinct structures. The double helix of DNA can be seen spiraling in its elegant dance. We can watch proteins fold, observe viruses binding to cells, witness the architecture of matter itself. Each improvement in microscopy has revealed layers of complexity we never suspected, and each revelation suggests that there are deeper layers still waiting, mysteries nested within mysteries, all the way down to the quantum foam, where reality itself becomes uncertain. That's 1/10,000th of a micrometer. To comprehend this scale requires a journey through orders of magnitude that strain the imagination. A micrometer is already invisibly small, 1/1,000,000th of a meter, far smaller than anything the human eye can perceive. And yet we must divide that distance by 10,000 to reach the resolution of our most advanced microscopes. At this level, we are operating in a realm where the very concepts of size and distance begin to lose their everyday meaning. We are seeing the arrangement of atoms, the fundamental building blocks that make up everything we know. A single atom of gold is roughly 1/10 of a nanometer in diameter, a sphere of matter so small that billions upon billions would fit on the head of a pin. And yet even these atoms are mostly empty space, their mass concentrated in nuclei thousands of times smaller still, surrounded by clouds of electrons that exist as probabilities rather than positions. To descend into this microscopic realm is to leave behind the solid, predictable world of our senses and enter a domain where matter behaves in ways that seem more like dreams than physics, where particles tunnel through barriers and exist in two places at once, where observation itself changes what is observed. Dinoflagellates are microscopic organisms that make ocean waves glow at night. On certain warm summer evenings, in certain bays and beaches around the world, the ocean comes alive with light. Each breaking wave erupts in blue-green fire. Each paddle stroke through the water leaves a glowing trail like liquid starlight. Each splash and ripple sparkles with bioluminescent brilliance. The source of this magic is dinoflagellates, single-celled organisms that drift by the millions through the upper layers of the sea. They are equipped with a remarkable chemical system that produces light when disturbed, a cold fire that requires no heat and wastes no energy. The light comes from a reaction between a molecule called luciferin and an enzyme called luciferase, the same chemistry that fireflies use to signal through summer nights. During the day, dinoflagellates are invisible, just another component of the plankton that colors the water green. But when darkness falls and the water is disturbed by movement, they respond with their ancient defense, turning the ocean into a constellation of microscopic stars. Swimming through bioluminescent waters is like moving through a living galaxy, a reminder that wonder exists not only in the distant cosmos, but in the closest waters, in the smallest lives, in the invisible communities that have illuminated the seas for hundreds of millions of years. The light from dinoflagellates is a defense mechanism to ward off predators. The glow that so enchants human observers serves a much more practical purpose in the microscopic world of the plankton. When a dinoflagellate is jostled by a hungry copod or disturbed by a small fish, it flashes with light, a sudden beacon in the dark water. This flash serves as what scientists call a burglar alarm, attracting the attention of larger predators who might eat whatever creature was disturbing the dinoflagellate in the first place. The tiny organism cannot fight or flee, but it can call for help in the only language the ocean understands: light. Over millions of years, this strategy has proven effective enough to persist, encoded into the dinoflagellate's genes and passed down through countless generations. The beauty we perceive is a side effect, a consequence of survival tactics refined in an arms race between microscopic herbivores and the single-celled photosynthesizers they hunt. Nature cares nothing for our aesthetic appreciation, and yet it produces wonders anyway, as if beauty were not an accident, but an inevitable consequence of complexity. A pattern that emerges wherever life grows intricate enough to surprise itself. Tardigrades have 8 stubby legs and walk with a bear-like gait. When observed under high magnification, moving slowly across a slide, tardigrades display a locomotion that is almost endearing in its deliberate clumsiness. Each of their 8 legs ends in several claws, and they use these to grip whatever surface they're traversing, whether it's a strand of moss, a grain of sand, or the glass of a microscope slide. They lift their legs in an alternating pattern, left, right, in a rhythm that closely resembles the walking gait of bears and other large mammals. This similarity is why they earned their common name, water bears, for despite being separated by vast differences in scale, they share this fundamental pattern of movement, this 4-beat rhythm of walking that evolution has discovered works at nearly any size. Watching a tardigrade walk is watching patience made visible, a slow and unstoppable progress across a landscape that to the tardigrade is as vast as a continent. They do not hurry. They move with the confidence of creatures who have survived every catastrophe Earth has thrown at life, who have outlasted ice ages and mass extinctions, who will likely still be walking their slow walk long after humanity has vanished. Under a microscope, tardigrades look oddly cute and surprisingly expressive. There is something unexpectedly charming about the appearance of tardigrades when seen magnified hundreds of times. Their rounded, segmented bodies taper toward the front, where a small head region contains a mouth that can extend outward like a tube. Depending on the species, they may appear translucent, allowing you to see the internal workings of their digestive system as they process their microscopic meals of algae or bacteria. Some have pigmentation that gives them color, pale yellows or soft greens that make them seem almost whimsical. Their faces, if you can call them that, have a blunt simplicity that the human brain cannot help but interpret as expression, a quality of presence that makes them feel less like abstract microorganisms and more like tiny animals with personalities. Scientists who study tardigrades often speak of them with affection, an unusual warmth for research subjects that cannot possibly return the sentiment. Perhaps it is their resilience that endears them to us, or their awkward, determined walking, or simply the fact that they exist at the boundary between the microscopic and the visible, close enough to our own scale that we can imagine ourselves in their place, wandering through a world of moss and water droplets, patient and indestructible. Microscopic organisms live in moss, lichen, and the thin films of water on leaves. The green cushions of moss that grow on the north side of trees, the crusty patches of lichen that decorate rocks and bark, the glistening droplets that cling to leaves after rain. All of these places are entire ecosystems at the microscopic scale. Within the layered structures of moss, there are tiny pools of water trapped between leaves, and in these pools live communities of organisms as diverse as any pond. Tardigrades graze on algae, Rotifers spin through the water with rings of beating cilia. Nematodes wriggle through the spaces between cells. Bacteria coat every surface in invisible films. Even a single dewdrop on a blade of grass can contain hundreds of species, all coexisting in a sphere of water no larger than a pearl. These microhabitats are everywhere, scattered across the world in uncountable billions, each one a pocket universe with its own ecology, its own cycles of predation and symbiosis, its own slow dramas of survival. To walk through a forest is to move through a three-dimensional landscape of these microscopic worlds, stacked and nested and overlapping. Invisible nations that persist regardless of whether any larger eye notices them. You can find tardigrades in your own backyard. The wonder of the microscopic world is not locked away in exotic locations or deep ocean trenches. It exists in the most ordinary places, waiting to be discovered. If you have a patch of moss growing on a stone, a clump of lichen on a fence post, or even a sample of damp soil from a flower bed, you almost certainly have tardigrades living there. Collecting them is simple. Soak the moss or lichen in water for a few hours, then squeeze the water out into a small container and examine a drop under a microscope. With patience and a little luck, you will see them. Tiny, translucent creatures walking slowly across the field of view, their stubby legs moving in that characteristic bear-like rhythm. It is a profound experience, that first sighting, to realize that these ancient survivors have been living just outside your door all along, invisible companions in the familiar landscape of home. The microscopic world is not distant or inaccessible. It is immediate, intimate, present in every handful of earth and every breath of air, waiting only for the right lens to bring it into focus. Viruses exist that only infect other viruses. In the arms race of microscopic life, evolution has produced layers of predation that challenge our simple categories of living and non-living. Viruses themselves occupy a strange borderland. They are not quite alive in the traditional sense, for they cannot reproduce on their own and lack the machinery of metabolism that defines cellular life. They are genetic instructions wrapped in protein shells, hijackers that invade cells and force them to manufacture more viruses. And yet even viruses can be parasitized. In 2008, scientists discovered the first virophage, a virus that infects other viruses. These tiny parasites wait until a larger virus invades a cell, and then they inject their own genetic material into the virus factory, sabotaging the production line and forcing it to create virophages instead. It is parasitism within parasitism, a nesting of dependencies that reveals just how complex the microscopic world has become. The discovery of virophages expanded our understanding of viral ecology and hinted at an entire hidden dimension of interactions happening inside infected cells, a microscopic theater where the players are not even fully alive, yet compete with strategies as sophisticated as any predator stalking through a forest. These virus parasites are called virophages. The name itself is telling. A combination of virus and phage, the latter borrowed from bacteriophages, viruses that infect bacteria. Virophages are smaller than the viruses they parasitize, just as parasites in the visible world are often smaller than their hosts. They carry only a handful of genes, the bare minimum needed to hijack an already hijacked cell. The first virophage discovered was named Sputnik, found living alongside a giant virus called mammavirus inside an amoeba. When Sputnik is present, the production of mammavirus particles drops dramatically, and many of the virus particles that do get made are deformed and defective, sabotaged by the virophage's interference. This relationship has likely been going on for millions of years, an invisible war fought inside single-celled organisms, where genetic sequences are the weapons and cellular machinery is the contested territory. The existence of virophages reminds us that complexity is not the sole province of large organisms, that even the simplest entities can evolve intricate relationships, and that the struggle for existence plays out at every scale, down to the molecular machinery inside a hijacked cell. Virophages can protect host organisms by weakening larger viruses. In the complex web of microscopic interactions, enemies of enemies can become accidental allies. When a virophage sabotages a larger virus, it reduces the burden on the infected cell, sometimes allowing the host to survive an infection that would otherwise have been fatal. In this way, virophages act as a kind of inadvertent immune system, a microscopic defender that arrives not out of loyalty, but out of its own parasitic self-interest. Some scientists speculate that virophages may have played a role in the evolution of cellular life, that ancient virophages might have helped early cells survive viral attacks during the chaotic epochs when life was first learning to organize itself. The relationship is not cooperation in any conscious sense, but it demonstrates a principle that runs through all of ecology. In a world of competing interests, the same interaction can be parasitism from one perspective and protection from another. The microscopic world does not deal in simple categories of good and evil, helper and harmer, but in a tangle of relationships so intricate that every organism is simultaneously predator, prey, parasite, and host, all roles shifting depending on context and scale. The quantum vacuum at the microscopic scale is never truly empty. If you could somehow remove every particle of matter and energy from a region of space, creating a perfect vacuum, you might expect to find absolute nothingness. But quantum physics reveals a stranger truth. Even in the emptiest space possible, there is a seething activity, a constant bubbling of particle-antiparticle pairs that flash into existence and annihilate each other in fractions of a second. These virtual particles borrow energy from the vacuum itself, appear for the briefest instant allowed by the uncertainty principle, and then vanish, returning the borrowed energy as they disappear. This quantum foam is not a theoretical abstraction, but a measured reality, confirmed by subtle effects in particle physics experiments and astronomical observations. The vacuum is alive with potential, humming with a background energy that permeates all of space. At the microscopic level, emptiness is not empty, but filled with possibility, a reminder that even nothing is something, that the universe at its smallest scales refuses to be still, and that the void itself is dancing with invisible potential. Microscopic particles blink in and out of existence in empty space. These quantum fluctuations are one of the strangest predictions of modern physics, and yet they have been verified again and again through precise experiments. In the vacuum, pairs of particles appear from nowhere. One particle and its antimatter twin, called into being by the uncertainty that governs quantum mechanics. For a moment, almost too brief to conceive, they exist, and then they meet and annihilate, releasing the energy they borrowed back into the void. This happens everywhere, at all times, in every cubic centimeter of space, an invisible firework display of creation and destruction. In certain circumstances, these virtual particles can have real effects on the world we experience. They contribute to the slight shifts in energy levels of atoms, They exert pressure on closely spaced metal plates in an effect called the Casimir force, and they may even play a role in the expansion of the universe itself. The boundary between existence and non-existence is not a solid line, but a blur, a quantum haze where particles flicker in and out of reality, reminding us that the universe is far stranger than our everyday experience suggests. Proteins inside cells fold into impossibly complex three-dimensional shapes. Every protein begins its life as a simple chain, a string of amino acids linked together like beads on a necklace. But the moment this chain is released from the ribosome that assembled it, something remarkable happens. The chain begins to fold, twisting and coiling and bending back on itself, driven by the chemical properties of its amino acids. Hydrophobic regions cluster together, hiding from the watery environment. Hydrogen bonds form between distant parts of the chain, pulling them close. Within seconds, the linear string has collapsed into a precise three-dimensional structure, a complex sculpture of loops and sheets and spirals that looks nothing like its linear origin. This shape is not random. It is determined by the sequence of amino acids, by the information encoded in the gene that produced the protein. And this shape is critical, for the shape determines the function. A protein is not just a molecule, but a machine. And like any machine, its geometry is its destiny. Some proteins form channels through cell membranes. Others catalyze chemical reactions millions of times per second. Still others bind to specific molecules with perfect specificity, like keys fitting into locks. The folding of proteins is one of nature's most elegant solutions, a way to encode complex function in simple linear information. A single cell contains thousands of different types of proteins. If you could inventory all the proteins in just one human cell, you would find an astonishing diversity. Each protein has a specific job, a particular function that contributes to the overall life of the cell. There are structural proteins that form the cell's skeleton, maintaining its shape and providing tracks for internal transport. There are enzymes that build and break down molecules, driving the chemical reactions of metabolism. There are signaling proteins that receive messages from outside the cell and relay them inward. There are transport proteins that shuttle molecules across membranes. There are motor proteins that walk along filaments carrying cargo, defense proteins that protect against invaders, regulatory proteins that turn genes on and off. This molecular workforce operates continuously, each protein performing its specialized task in coordination with thousands of others, creating an emergent order that we call life. The fact that all of these proteins are built from the same 20 amino acids, just arranged in different sequences, is a testament to the power of combinatorial complexity. The way simple building blocks can generate infinite variation when assembled in different orders. Microscopic molecular motors walk along cellular highways carrying cargo. Inside every living cell, there is a transportation network more organized than any city's roads. Long filaments of protein stretch across the cell like highways, and along these highways walk molecular motors, proteins that literally take steps. Kinesin is one such motor, a protein with two leg-like extensions that walk along microtubules, tubular structures that crisscross the cell. Attached to the kinesin's back is cargo, a vesicle full of neurotransmitters, a mitochondrion that needs to be delivered to a distant part of the cell, a packet of proteins destined for the membrane. The kinesin walks step by step, powered by ATP, the cellular fuel, each step precisely 8 nanometers long. It walks with a rhythm, left, right, just like the tardigrade walking through moss, just like a bear walking through a forest. the same mechanical solution to the problem of movement, discovered independently at scale separated by factors of millions. These motors are essential to life. Without them, cells would be pools of mixed components with no organization, no ability to concentrate resources where needed, no way to maintain the compartmentalization that allows complex chemistry to occur without interference. These motors take literal steps, 1 nanometer at a time. The walking of kinesin is not a metaphor, but a physical reality, confirmed by sophisticated microscopic techniques. Scientists have watched individual motor proteins as they march along their filament tracks, and the motion is unmistakably step-like. The protein has 2 domains that act as feet, and it alternates between them, lifting one while the other remains planted, then swinging the lifted foot forward to take the next step. Each step is precisely measured, determined by the structure of the tubule along which it walks. The distance is 8 nanometers, a span so small that you could fit 125 million of these steps into a single meter. And yet the step is consistent, reliable, repeated billions of times without error. The motors move at a pace that seems agonizingly slow when scaled to human dimensions. Yet within the microscopic environment of the cell, they are remarkably efficient, capable of transporting cargo from one end of a neuron to the other, a journey that at cellular scale is like crossing a continent. In their patient walking, we see another echo of the universal patterns of motion, the same solutions appearing again and again across the scales of existence. Mitochondria are microscopic powerhouses inside every cell. Floating through the cytoplasm of nearly every cell in your body are small oblong structures called mitochondria, and they are responsible for converting the food you eat and the oxygen you breathe into usable energy. They are the power plants of the cell, taking in sugar and oxygen and producing ATP, the molecular currency that fuels all cellular work. A single cell might contain hundreds or even thousands of mitochondria, depending on how much energy that cell needs. Muscle cells, which require enormous amounts of power to contract, are packed with mitochondria. Brain cells, which burn through energy maintaining the electrical signals of thought, are similarly dense with these organelles. Under an electron microscope, mitochondria reveal an intricate internal structure, a smooth outer membrane and a folded inner membrane that creates vast surface area for energy production. The folds, called cristae, are lined with the molecular machinery that pumps protons, creating a gradient that drives the synthesis of ATP like water turning a turbine. Without mitochondria, complex multicellular life as we know it could not exist, for they provide the concentrated energy needed to power the intricate chemistry of cells that do more than simply survive, but grow, communicate, move, and think. Mitochondria have their own DNA, separate from the cell's nucleus. One of the most fascinating aspects of mitochondria is their independence. Unlike most cellular structures, which are built according to instructions from the DNA in the nucleus, mitochondria carry their own genetic material, a small circular loop of DNA that resembles the chromosomes of bacteria. This DNA codes for some, though not all, of the proteins that mitochondria need to function. The rest are imported from the cell, created by the nucleus and delivered to the mitochondrion like supplies sent to an outpost. This arrangement is not accidental, but historical. The prevailing scientific theory holds that mitochondria were once free living bacteria, similar to modern purple bacteria, that were engulfed by a larger cell billions of years ago. Instead of being digested, they survived inside their host and began a symbiotic relationship that has persisted across evolutionary time. The host cell provided protection and nutrients, while the mitochondria provided energy. Over millions of years, the two became inseparable, their fates intertwined, until the mitochondria could no longer survive outside the cell, and the cell could no longer survive without the mitochondria. Every breath you take, every movement you make, depends on this ancient partnership, a merger of two forms of life that occurred in the distant microscopic past. Red blood cells are so small that 5 million fit in a single drop of blood. If you could magnify a drop of blood to the size of a swimming pool, you would find yourself surrounded by 5 million disc-shaped cells, each one slightly compressed in the middle, like a pillow that has been squeezed. These are red blood cells, and they have a singular purpose: to carry oxygen from your lungs to every tissue in your body, and to return carbon dioxide for exhalation. They are among the simplest cells in the human body, so specialized for their task that they have discarded their nuclei and most other organelles, becoming little more than sacs of hemoglobin, the protein that binds oxygen and gives blood its red color. Their small size allows them to slip through the narrowest capillaries, vessels so tiny that the red cells must fold and squeeze to pass through single file. Their lifespan is brief, only about 120 days, after which they are filtered out by the spleen and recycled. Every second, your body produces and destroys millions of red blood cells, maintaining a constant population that flows endlessly through the circulatory system. A river of oxygen delivery that never stops, never rests, sustaining every moment of consciousness and every heartbeat from birth until death. White blood cells hunt bacteria like microscopic predators. While red blood cells flow passively through the bloodstream, white blood cells are active hunters. They patrol the body searching for signs of infection, and when they find bacteria or other invaders, they attack with strategies that are surprisingly sophisticated. Some white blood cells, called neutrophils, are the first responders to infection. They squeeze through the walls of blood vessels and follow chemical trails to the site of invasion, moving through tissue with determination. When they encounter a bacterium, they extend portions of their membrane around it, engulfing the invader in a process called phagocytosis. The bacterium is drawn inside the white blood cell into a compartment called a phagosome, which then fuses with lysosomes filled with digestive enzymes and toxic chemicals. The bacterium is dissolved, broken down into harmless components that are either recycled or expelled. Time-lapse microscopy of this process reveals behavior that looks strikingly like hunting. The white cell senses its prey, pursues it, captures it, and consumes it, all at the microscopic scale, all driven by molecular signals rather than conscious intent. In these tiny battles, fought by the billions inside your body every day, we see the same patterns of predation that exist in every ecosystem, from the ocean to the savanna. Proof that the logic of survival operates at every level of life. Dust particles floating in sunbeams are ecosystems for microscopic life. When sunlight streams through a window and illuminates the air, you can see them. Countless tiny specks drifting lazily through the beam, rising and falling on invisible currents. These particles seem insignificant, mere bits of lint and dead skin. But at the microscopic level, they are entire worlds. Each dust particle is a raft carrying bacteria, fungal spores, pollen fragments, and microscopic mites, all traveling together through the atmosphere. Some bacteria can remain dormant on these particles for months or years, waiting for conditions favorable enough to resume their growth. The spores of molds and fungi ride the dust like seeds on the wind, dispersing to new locations where they might germinate and colonize. Even viruses can attach themselves to dust particles, hitchhiking through the air from one host to another. The dust that settles on furniture and collects in corners is not inert matter, but a repository of microscopic travelers, a snapshot of all the invisible life that shares our spaces. To watch dust drift through a sunbeam is to watch an aerial migration of the very small, a quiet caravan of microorganisms making their slow way through the air we breathe. Pollen grains have intricate geometric patterns visible only under magnification. In spring, when plants release their pollen to the wind or to visiting insects, they are launching billions of microscopic capsules, each one a masterpiece of natural engineering. Under a microscope, pollen grains reveal themselves as astonishingly ornate structures. Some are covered in spikes like tiny medieval maces. Others have patterns of grooves and ridges as regular as circuit boards. Still others are studded with knobs or decorated with intricate lacework. These patterns are not decorative, but functional, designed to help the pollen stick to pollinators or to survive the journey through the air. The outer wall of a pollen grain, called the exine, is made of one of the most durable biological materials known, resistant to decay and capable of surviving for millions of years in the fossil record. Each grain is a genetic delivery vehicle, carrying the male reproductive cells of the plant wrapped in this tough, beautiful armor. The diversity of pollen shapes is so great that scientists can identify plant species by their pollen alone, each type as distinctive as a fingerprint, each one a work of microscopic art shaped by millions of years of evolution. Each species of plant has uniquely shaped pollen. If you were to collect pollen from a dozen different plants and examine them under a microscope, you would find that no two species produce identical grains. Grasses release smooth, spherical pollen. Pine trees produce pollen with air bladders that help it float on the wind. Orchids make pollen clumped together in packets called pollinia. Dandelions have spiky grains that catch easily on fur and clothing. This diversity is the result of each plant adapting its pollen to its particular method of reproduction and its relationship with pollinators. Flowers pollinated by bees tend to have sticky, textured pollen that adheres easily to the bee's fuzzy body. Wind-pollinated plants produce smooth, light pollen in enormous quantities, counting on sheer numbers to ensure that some grains find their target. The shape of a pollen grain tells the story of the plant's reproductive strategy, written in microscopic sculpture, a text that scientists can read to understand not only the present ecology of a region, but also its ancient past. For fossilized pollen preserved in sediment reveals which plants grew thousands or millions of years ago, a microscopic archive of botanical history. Snowflakes form microscopic ice crystals in endlessly varying patterns. When water vapor in a cloud freezes around a tiny particle of dust or pollen, a snowflake begins its life. The ice crystal that forms is not random, but follows the hexagonal symmetry dictated by the molecular structure of water. As the crystal falls through the cloud, it encounters varying conditions of temperature and humidity, and these conditions determine how it grows. In some conditions, the crystal extends its 6 arms into long needles. In others, it develops intricate branches and side branches, creating the classic star shape we associate with snowflakes. Still, other conditions produce flat plates, hollow columns, or combinations of forms so complex they defy simple description. Each snowflake's journey through the cloud is unique, and because the growth pattern is so sensitive to environmental conditions, each snowflake develops its own distinct personality, a frozen record of the path it took through the sky. Under magnification, snowflakes are revealed as structures of breathtaking delicacy and geometric precision. Nature's most ephemeral sculptures, crystals of ice that exist for mere moments before melting, their patterns dissolving back into formless water. No two snowflakes have ever been exactly identical at the molecular level. The oft-repeated claim that no two snowflakes are alike is more than poetic fancy. It is a statistical reality rooted in the mathematics of complexity. The number of possible arrangements of water molecules in a snowflake is astronomically large, far exceeding the number of snowflakes that have ever fallen in all the winters of Earth's history. Each tiny variation in temperature, each slight change in humidity, each microscopic difference in the crystal's path through the cloud creates a different pattern of growth. At the molecular level, the uniqueness becomes absolute. Even if two snowflakes appeared identical under a microscope, the precise positions of their individual water molecules would differ. For the quantum uncertainty governing molecular positions ensures that perfect duplication is impossible. This uniqueness is both humbling and beautiful, a reminder that variation is written into the fabric of nature, that the universe tends toward diversity rather than repetition, and that every small thing, every falling crystal, every microscopic structure is in some sense irreplaceable, a singular event in the vast unfolding of time. Salt crystals grow in perfect cubic structures at the microscopic scale. When seawater evaporates from a tidal pool, or when you sprinkle table salt into a dish and let the water dry, what remains are crystals of sodium chloride, and each one is a tiny monument to mathematical perfection. Under magnification, salt crystals reveal themselves as perfect cubes, their edges straight, their angles exactly 90 degrees, their faces smooth and flat. This shape is not imposed from outside, but emerges naturally from the arrangement of sodium and chloride ions, which stack together in an alternating three-dimensional lattice. Each sodium ion is surrounded by 6 chloride ions, and each chloride ion is surrounded by 6 sodium ions. And this repeating pattern extends outward in all directions, building the crystal layer by layer, molecule by molecule, following rules encoded in the electrical charges that hold the structure together. The crystal grows as more ions attach to its surfaces, always maintaining the cubic geometry, always extending the pattern outward with perfect fidelity. In a handful of salt, there are millions of these microscopic cubes, each one identical in principle, yet unique in size and detail, an entire geometry lesson crystallized from the sea. Sugar crystals form in beautiful geometric arrangements. When you dissolve sugar in hot water and then allow the solution to cool slowly, the sugar molecules begin to find each other again, assembling into solid crystals that emerge from the liquid like architecture growing from nothing. Sugar crystals favor a different geometry than salt, forming shapes that are more elongated, with angled faces and pointed ends. Under a microscope, they reveal a complexity of structure, with layers and terraces visible on their surfaces where molecules have stacked unevenly. Some sugar crystals grow into long prisms, others into blocky shapes, still others into intricate clusters that branch and intersect. The particular shape depends on the conditions under which the crystal forms, the temperature and concentration, and the presence of impurities. But underlying all this variation is the same fundamental pattern, the same arrangement of carbon, hydrogen, and oxygen atoms locked into repeating units. Rock candy, the sweet crystals that grow on strings in sugar solutions, are large enough to see the structure with the naked eye. But even the smallest grain of granulated sugar contains this hidden order, this microscopic testimony to the fact that molecules, given the chance, will organize themselves into patterns of surprising regularity and beauty. Spider silk is made of microscopic protein strands stronger than steel. When a spider spins its web, pulling silk from its spinnerets with a delicate movement of its legs, it is creating one of nature's most remarkable materials. The silk emerges as a liquid protein that solidifies the moment it contacts the air, forming a fiber thinner than a human hair, yet possessing strength that exceeds high-grade steel when compared by weight. Under electron microscopy, spider silk reveals its secret. It is composed of protein molecules arranged in parallel chains that form crystalline regions interspersed with more flexible, amorphous sections. This combination gives silk both strength and elasticity, allowing it to absorb the impact of a flying insect without breaking, stretching to accommodate the shock, and then contracting back to its original length. The proteins that make up silk contain repeating sequences of amino acids, patterns like musical motifs that determine how the molecules fold and align. Different types of silk for different purposes. The sticky capture spiral of a web, the strong support threads, the protective silk that wraps eggs, are made from slightly different proteins, each optimized for its task. In the microscopic architecture of spider silk, we find elegance and efficiency combined, a material so well designed that engineers are still struggling to replicate it in laboratories. The microscopic structure of butterfly wings creates their iridescent colors. When you see a butterfly flash brilliant blue or shimmer with metallic green as it flits through a garden, you are witnessing color created not by pigments, but by physics. The wings of butterflies are covered in thousands of microscopic scales, and these scales have elaborate three-dimensional structures that interact with light in complex ways. Some scales have layers separated by precise distances, and when light reflects off these layers, the reflected waves interfere with each other, cancelling out some wavelengths and reinforcing others. This is the same principle that creates the rainbow shimmer in soap bubbles or the colors in an oil slick. Other scales have ridges and grooves arranged like diffraction gratings, splitting light into its component colors. Still others have elaborate tree-like structures called photonic crystals that reflect only specific wavelengths while allowing others to pass through. The result is color that changes depending on the angle of viewing, an iridescence that seems to glow from within, more vibrant and pure than any pigment could produce. Under magnification, these structures reveal themselves as astonishingly intricate, tiny machines for manipulating light, proof that evolution has discovered the principles of optics and nanofabrication long before humans did, and has deployed them with a beauty that our technology still struggles to match. Butterfly wing scales are arranged like overlapping roof tiles. If you could magnify a butterfly wing to the size of a billboard, you would see that its surface is not smooth, but covered in thousands upon thousands of tiny scales, each one a flattened structure shaped like an elongated teardrop or a miniature shingle. These scales overlap each other in regular rows, like tiles on a roof or feathers on a bird, creating a surface that is both flexible and protective. Each scale is attached to the wing membrane by a tiny stalk, and the scales can move independently, which gives the wing its velvety texture and helps it shed water. The scales are made of chitin, the same material that forms the butterfly's exoskeleton, and they are incredibly light, contributing almost no weight to the wing despite their numbers. When you handle a butterfly and see colored dust on your fingers, you are seeing these scales dislodged from the wing and carried away. Without them, the wing is transparent, a delicate membrane with a network of veins for support. The scales serve multiple purposes. They create color, they provide insulation, they help the butterfly shed rain, and they may even play a role in courtship displays. In their orderly arrangement, we see nature's solution to the challenge of creating a surface that is light, strong, colorful, and waterproof all at once. Peacock feathers don't contain blue pigment. Microscopic structures create the color through light interference. The dazzling blue-green of a peacock's display is not created by any dye or pigment molecule, but by the microscopic architecture of the feather itself. Each barb contains layers of keratin separated by air pockets, and these layers are spaced at precise distances that reflect blue and green wavelengths while cancelling out others. The color shifts as the peacock moves, changing from emerald to sapphire to turquoise, depending on the angle of light and observation. This structural color is more brilliant and more durable than any pigment, never fading as long as the microscopic structure remains intact. A peacock's call to beauty, written in physics rather than chemistry. The microscopic hooks on a gecko's feet allow it to walk on ceilings. When a gecko walks upside down across a ceiling or climbs a vertical pane of glass, it is using one of nature's most ingenious adhesion systems, one that requires no glue, no suction, and no moisture. Each toe pad is covered in millions of microscopic hairs called setae, and each seta branches into hundreds of even tinier tips called spatulae. These spatulae are so small and so numerous that they can interact with surfaces at the molecular level through weak forces called van der Waals interactions. Individually, each bond is almost negligible, but multiplied across millions of contact points, the combined force is strong enough to support the gecko's entire weight. The gecko can release its grip simply by changing the angle of its foot, peeling the setae away without effort, a reversible adhesion that has inspired engineers seeking to create climbing robots and reusable adhesive tapes. Each gecko footpad has millions of microscopic hairs. The density of these structures is staggering, approximately 14,000 setae per square millimeter, each one only a few micrometers in diameter, invisible to the naked eye. Together, these millions of hairs create a surface area so vast that a single gecko foot, if all its setae were in contact with the surface, could theoretically support the weight of a small child. The gecko uses only a fraction of this adhesive capacity in normal climbing, giving it a comfortable safety margin as it scurries across surfaces where any other creature its size would fall. It is a solution born from the microscopic scale, where surface area dominates over weight, and where forces too weak to matter at human dimensions become powerful enough to defy gravity. Plant roots are covered in microscopic root hairs that absorb water. When you pull a seedling from soil and rinse away the dirt, the root appears smooth and simple, but under magnification, it reveals itself as densely fuzzy, covered in thousands of microscopic extensions called root hairs. Each hair is a single cell that has grown outward from the root surface, extending into the spaces between soil particles where water and dissolved minerals collect. These hairs vastly increase the surface area available for absorption, allowing the plant to extract water and nutrients far more efficiently than a smooth root could. They grow continuously at the root tip as it pushes through the soil, and they die and are shed behind, replaced by new hairs in an endless cycle. In their delicate microscopic reach, we see how plants explore the soil, sending out thousands of tiny fingers to feel for resources in the dark underground world. A single plant root can have billions of microscopic hairs. A mature plant, even a small one, may have a root system that extends for meters through the soil, and every centimeter of that system is covered in these absorbing hairs. The total surface area created by all these microscopic extensions can exceed the area of the plant's leaves by many times, a hidden vastness devoted entirely to gathering water from the earth. This is why transplanting can be so traumatic for plants. The delicate root hairs are easily damaged, and the plant must regrow them before it can resume normal water uptake. The root hair is the plant's connection to the soil, its microscopic interface with the mineral world, and without these billions of tiny structures, life on land as we know it could not exist. Fungi spread through soil using microscopic threads called hyphae. When you see a mushroom emerging from the ground, you are seeing only the fruiting body, the reproductive structure of a much larger organism hidden beneath the surface. The main body of the fungus consists of hyphae, thin threads of cells that grow through the soil in all directions, branching and rebranching into a network of almost unimaginable complexity. A single hypha is only a few micrometers in diameter, thin enough to slip between soil particles and penetrate decaying wood. As hyphae grow, they secrete enzymes that break down organic matter, digesting it externally and absorbing the nutrients. The network expands slowly, sometimes covering areas of many square meters, all connected, all part of a single organism. In the autumn, when conditions are right, some hyphae gather together and form the mushrooms we see, but the vast majority of the fungus remains invisible, a microscopic web beneath our feet. Mycelium networks connect entire forests underground at the microscopic level. The hyphae of fungi do not grow in isolation, but form vast interconnected webs called mycelium. And these networks link tree to tree across entire forests. The fungi form partnerships with tree roots, wrapping around them and even penetrating the root cells. Through this connection, nutrients flow in both directions. The fungus provides the tree with minerals and water extracted from the soil, while the tree provides the fungus with sugars produced through photosynthesis. But the network does more than simply feed individual trees. It acts as a communication system, allowing trees to share resources and even warning signals. A tree under attack by insects can send chemical signals through the mycelium to neighboring trees, which then prepare their defenses. This microscopic web, invisible and silent, is the forest's hidden nervous system. Trees communicate through microscopic fungal networks. The discovery that trees are not isolated individuals, but members of a connected community, has transformed our understanding of forests. Through the mycelial threads that link root to root, a mother tree can send excess carbon to her shaded seedlings, giving them the energy they need to survive until they reach sunlight. Dying trees have been observed redistributing their resources to their neighbors before they succumb, a final gift passed through the microscopic highway beneath the soil. This communication happens at the molecular level, chemical messages traveling through fungal cells too small to see, yet the effect is profound. shaping the health and resilience of entire ecosystems through connections measured in micrometers. Microscopic bacteria in soil help plants absorb nutrients. The soil is not merely dirt, but a living matrix, teeming with bacteria that perform essential services for plant roots. Some bacteria convert atmospheric nitrogen into forms that plants can use, a process called nitrogen fixation. Others break down organic matter, releasing phosphorus and other minerals. Still others form protective films around roots, defending against pathogens. These microscopic partners are so essential that plants grown in sterilized soil, free of bacteria, struggle to thrive even when provided with water and minerals. The microscopic world of the soil is what transforms rock and decay into fertility, what makes growth possible in the thin layer of earth where roots and microbes meet. The smell of rain comes from microscopic bacteria in the soil. That distinctive earthy scent that rises when rain falls on dry ground has a name: petrichor, and its source is microscopic. Bacteria in the soil, particularly a group called actinomycetes, produce a chemical compound called geosmin as part of their life cycle. This molecule sits on the soil particles and inside the bodies of the bacteria, and when raindrops hit the ground, they release it into the air along with tiny aerosol particles. Geosmin is one of the most potent scent molecules known. The human nose can detect it at concentrations as low as 5 parts per trillion. The smell of rain is actually the smell of microscopic life awakening, bacteria responding to moisture after drought, releasing their ancient chemical signature into the air we breathe. Microscopic phytoplankton produce more than half the oxygen we breathe. Every other breath you take comes from the ocean, from organisms so small that millions fit in a teaspoon of seawater. These are the phytoplankton, microscopic algae and bacteria that drift through the upper layers of the sea, capturing sunlight and using it to split water molecules and release oxygen as a byproduct. Cyanobacteria, diatoms, dinoflagellates, and countless other species perform this photosynthesis continuously, day after day, across all the oceans of the world. Their collective productivity exceeds that of all terrestrial plants combined, making them the primary source of oxygen for the planet. We owe every breath to these invisible drifters, to the microscopic green cells floating in waters we may never see. Phytoplankton are so small that millions fit in a teaspoon of seawater. A single drop of ocean water, no larger than what might fall from an eyedropper, contains an entire ecosystem of photosynthetic life. These microscopic organisms are the foundation of the marine food web, eaten by zooplankton, which are eaten by small fish, which are eaten by larger fish, all the way up to whales and sharks. The abundance of life in the ocean, from sardines to blue whales, ultimately depends on these microscopic photosynthesizers, on their ability to turn sunlight and dissolved carbon dioxide into living tissue. They are proof that size is no measure of importance, that the smallest organisms can be the most essential. Coral polyps are microscopic animals that build massive reef structures. The Great Barrier Reef, visible from space, is built by creatures barely larger than the head of a pin. Individual coral polyps are tiny, soft-bodied animals related to jellyfish and sea anemones. Each polyp secretes a skeleton of calcium carbonate beneath itself, building a small cup in which it lives. As generations of polyps live and die, their skeletons accumulate, layer upon layer, slowly constructing the massive limestone structures we call coral reefs. A reef that seems solid and permanent is actually a colony of millions of microscopic animals, each one contributing its small effort to a construction project that spans centuries. In their patient building, we see how the microscopic and the monumental are connected, how small actions accumulate into transformations visible across oceans. A single coral is actually a colony of thousands of microscopic organisms. What appears to be one coral head is in fact a community, thousands of individual polyps living together in coordinated harmony. They share a connected tissue layer and coordinate their feeding, extending their tentacles at night to capture microscopic prey from the water. Each polyp is both independent and interdependent, able to feed itself, yet contributing to the collective structure. The colony grows as polyps divide, creating new individuals that remain attached to their parents, building outward in patterns determined by species and environment. It is a form of life that blurs the boundary between individual and community, where the question, what is an organism, becomes surprisingly difficult to answer. The colors in coral come from microscopic algae living in their tissues. The vibrant yellows, greens, and browns of living coral are not produced by the coral polyps themselves, but by microscopic algae called zooxanthellae that live inside the coral's transparent tissues. These algae perform photosynthesis, using sunlight to create sugars that they share with their coral hosts. In return, the coral provides the algae with a safe home and access to sunlight. This partnership is so essential that without their algae, most corals cannot survive. When corals are stressed by heat or pollution, they expel their algae, turning ghostly white in a process called bleaching. The coral is not dead but starving, and unless conditions improve and the algae return, the polyps will eventually die, leaving only the white skeleton behind. Sea foam is created by proteins from microscopic plankton. When waves crash on a beach and leave behind drifts of foam that look like clouds fallen to earth, they are creating something from the invisible remains of microscopic life. As phytoplankton die and decompose, they release proteins and other organic compounds into the seawater. When waves agitate this protein-rich water, the proteins act like soap, stabilizing bubbles and creating foam that can pile up in thick layers. The foam is temporary, collapsing as the proteins break down further, or as the water drains away. But for a brief time, it transforms the boundary between sea and land into something ethereal and strange, a monument to the invisible organisms whose bodies have dissolved into the chemistry of the ocean. Ice cream gets its smooth texture from microscopic ice crystals. The pleasure of eating ice cream lies partly in its texture, and that texture depends on keeping ice crystals microscopic. When ice cream is frozen quickly and stored at very cold temperatures, the water in the mixture forms tiny crystals too small to detect on the tongue. If ice cream melts and refreezes slowly, those crystals grow larger, creating a grainy, unpleasant texture. Premium ice cream is smoother because it contains less air and more fat, both of which interfere with crystal growth, keeping them small and innumerous. The creaminess we taste is actually the absence of sensation. The crystals so microscopic that they slip past our perception, leaving only the impression of smoothness. Bread rises because of microscopic yeast consuming sugar and releasing gas. When dough sits in a warm place and slowly swells, growing soft and airy, it is being transformed by billions of yeast cells eating and breathing. The yeast metabolize the sugars in the flour, breaking them down through fermentation and releasing carbon dioxide as waste. This gas forms bubbles in the elastic dough, which stretches to accommodate them, causing the whole mass to expand. Each bubble is a tiny monument to microscopic respiration, to the ancient partnership between humans and single-celled fungi that has given us bread for thousands of years. Each bubble in bread is a tiny monument to microscopic life. When you slice through a loaf and see the intricate structure of holes and chambers, you are seeing the architecture created by yeast, the spaces where carbon dioxide collected and expanded during proofing and baking. The heat of the oven eventually kills the yeast, but their work remains, frozen in the structure of the baked bread. Without these microscopic organisms, bread would be dense and hard, closer to a cracker than the soft, airy loaves we know. Every sandwich, every piece of toast, every dinner roll exists because of the invisible labor of yeast. Cheese develops flavor from microscopic bacteria and molds. The transformation of milk into cheese is a slow conversation between humans and microorganisms, a partnership refined over millennia. Bacteria acidify the milk, causing proteins to coagulate. Rennet enzymes separate curds from whey. As the cheese ages, bacteria and molds continue their work, breaking down proteins and fats into smaller, more flavorful compounds. Different species of microorganisms create different flavors. The blue veins in Roquefort are mold. The holes in Swiss cheese are created by bacteria that produce carbon dioxide. The sharp tang of aged cheddar comes from bacterial enzymes working over months or years. The microscopic residents of cheese are what make each variety unique. Yogurt is created by microscopic bacteria fermenting milk. The thick, tangy texture of yogurt is the work of bacteria, primarily Lactobacillus and Streptococcus species, which consume the lactose in milk and produce lactic acid. This acid causes the milk proteins to coagulate and thicken, transforming liquid into the creamy substance we know as yogurt. The bacteria remain alive in the finished product, billions of them in every spoonful, continuing their microscopic existence in the cool darkness of refrigeration until consumed. Cultures of these bacteria have been passed from generation to generation for thousands of years. Microscopic heirlooms that connect us to ancient methods of preserving food. The microscopic bacteria in your gut help you digest food. Your intestines are home to trillions of bacteria that form a complex ecosystem more diverse than a rainforest. These microorganisms break down dietary fibers that your own enzymes cannot process, extracting nutrients and energy from food that would otherwise pass through undigested. They synthesize vitamins, particularly B vitamins and vitamin K, providing essential nutrients your body cannot make on its own. This microscopic community is so integral to digestion that without it, nutrition itself becomes compromised, a reminder that we are never truly independent, but always in partnership with invisible allies. Your gut contains trillions of microscopic organisms. The human gut microbiome contains approximately 100 trillion bacterial cells, outnumbering human cells and creating a complex ecosystem that weighs several pounds. This population includes thousands of different species, each occupying its own niche within the landscape of the intestines. Some cluster near the stomach where conditions are more acidic. Others thrive in the oxygen-poor depths of the colon. They compete and cooperate, forming communities that remain relatively stable over time, yet can shift in response to changes in diet, medication, or stress. The gut is not merely a digestive tube, but a living habitat, a microscopic wilderness within. These gut microbes can influence your mood and emotions. The connection between gut bacteria and brain function, sometimes called the gut-brain axis, is one of the most surprising discoveries of modern microbiology. Gut bacteria produce neurotransmitters like serotonin and dopamine, the same chemicals that regulate mood in the brain. They communicate with the nervous system through the vagus nerve, sending signals that can affect anxiety, depression, and even behavior. Studies have shown that altering the gut microbiome can change emotional responses in both animals and humans, suggesting that these microscopic organisms play a role in shaping not just physical health, but mental states as well. Microscopic organisms in your gut produce vitamins your body needs. Among the many services provided by gut bacteria, vitamin synthesis is particularly crucial. Bacteria in the colon produce vitamin K, essential for blood clotting, and several B vitamins, including biotin, folate, and B12. Without these microscopic factories working continuously in the darkness of the intestines, vitamin deficiencies would develop despite adequate dietary intake. The partnership between human and bacteria is so ancient that our bodies have come to depend on it, outsourcing certain biochemical functions to our microscopic residents. Human skin is home to millions of microscopic mites. Living in the pores and hair follicles of nearly every human being are tiny arthropods called demodex mites, creatures with elongated bodies and 8 stubby legs. They spend their entire lives on human skin, emerging at night to mate and then retreating back into follicles before dawn. They feed on sebum, the oily secretion of skin glands, and on dead skin cells. Most people never know these mites exist, for they cause no symptoms and go about their lives in peaceful coexistence with their hosts, a microscopic fauna that has evolved alongside humanity for so long that we are their only habitat. These skin mites are harmless and help keep skin clean. Far from being parasites in the harmful sense, demodex mites may actually benefit their hosts by consuming excess oils and dead cells, acting as microscopic janitors that keep pores from becoming clogged. They are so ubiquitous that scientists consider them part of the normal skin ecosystem, present on virtually all adult humans regardless of hygiene or geography. Their presence is a reminder that we are not individuals but ecosystems, that the boundary of self extends to include countless other organisms living on and within us. Every surface in your home hosts invisible microscopic ecosystems. Kitchen counters, bathroom sinks, doorknobs, and floors all support communities of bacteria, fungi, and other microorganisms. Each surface with its own characteristic population shaped by moisture, temperature, and the frequency of human contact. These communities are mostly harmless, composed of environmental organisms and microbes from human skin. Some surfaces, like cutting boards, require regular cleaning to prevent harmful bacteria, but many surfaces harbor only benign residents. The microscopic world does not stop at our doors, but flows through our homes, adapting to the environments we create. The air you breathe contains thousands of microscopic particles and organisms. Each breath draws in not just oxygen and nitrogen, but an invisible cargo of dust, pollen, bacterial spores, fungal fragments, and living microorganisms suspended in the atmosphere. Some of these particles are so small they remain airborne for hours or days, drifting on currents too subtle to feel. Others are larger, settling slowly toward the ground. The air is not empty, but full, a medium through which the microscopic world travels, dispersing and colonizing, moving from place to place on invisible winds. Every inhalation is a sampling of this aerial ecosystem, a reminder that we are continuously immersed in the microscopic dimension. Clouds form around microscopic particles of dust, salt, or smoke. Water vapor in the atmosphere cannot simply condense into droplets on its own. It needs a surface to gather upon. Tiny particles called cloud condensation nuclei provide these surfaces, allowing water molecules to collect and form the droplets that make up clouds. These nuclei can be dust lifted from deserts, salt crystals from ocean spray, or smoke particles from wildfires. Each cloud droplet begins with a single microscopic particle at its core, a speck of matter that becomes the seed for something visible. Proof that the largest weather systems begin at the smallest scales. Each raindrop begins as a microscopic droplet. High in a cloud, water condenses around a nucleus, forming a droplet so small it floats rather than falls. As more water vapor collects, the droplet grows, colliding with other droplets and merging with them. Only when it reaches sufficient size does gravity overcome air resistance, and the droplet begins its descent as rain. By the time it strikes your face or a leaf or the ground, it has grown millions of times larger than its microscopic beginning, yet it carries that origin within it still, the particle around which everything else gathered. Fog is made of microscopic water droplets suspended in air. When you walk through fog, you are moving through a cloud at ground level, surrounded by water droplets so small they cannot fall, but instead drift in the air like smoke. Each droplet is only 10 to 20 micrometers in diameter, far too small to see individually, yet collectively, they reduce visibility to almost nothing. The dampness you feel is these droplets accumulating on your skin and clothing. Thousands upon thousands of microscopic spheres settling on surfaces. Fog is the microscopic world made visible, a demonstration that what seems empty is actually full. Dew forms as microscopic droplets condense on cool surfaces. On a clear night, surfaces radiate heat into space and cool below the temperature of the surrounding air. Water vapor touching these cool surfaces condenses, first as microscopic droplets too small to see, then growing and merging until they become the visible beads we call dew. Each drop is a collection of countless microscopic condensation events, water molecules slowing from their gaseous dance and settling into liquid stillness. Morning dew is the microscopic process of phase transition made manifest, a nightly reminder of water's constant movement between states. Frost creates microscopic ice patterns on windows. When temperatures drop below freezing and water vapor contacts a cold window, it transforms directly from gas to solid, bypassing the liquid phase in a process called deposition. The ice crystals that form grow in intricate patterns, branching fractals that spread across the glass like frozen ferns. At the microscopic level, each branch is an ordered arrangement of water molecules locked into crystalline structure, the same hexagonal geometry that governs snowflakes. The patterns are unique to each window each night, shaped by temperature gradients and imperfections in the glass, yet all follow the same underlying rules encoded in the physics of ice. The blue in the sky comes from microscopic air molecules scattering sunlight. When sunlight enters the atmosphere, it encounters molecules of nitrogen and oxygen, particles so small they are measured in nanometers. These molecules scatter shorter wavelengths of light more effectively than longer wavelengths, and blue light has a shorter wavelength than red. The scattered blue light reaches your eyes from all directions, creating the impression of a blue dome overhead. At sunset, when sunlight travels through more atmosphere, even more blue light is scattered away, leaving the longer red wavelengths to paint the sky. The color of the sky itself is a microscopic phenomenon. Light interacting with particles too small to see, creating the backdrop against which all visible life unfolds. Microscopic organisms can survive in boiling hot springs, in volcanic regions where water emerges from the earth at temperatures that would instantly kill most life. There are bacteria and archaea that not only survive but thrive. These thermophiles have evolved proteins and membranes that remain stable at temperatures exceeding 100 degrees Celsius, dwelling in waters hot enough to cook an egg. They extract energy from minerals dissolved in the superheated water, living in conditions that resemble the early Earth when life first began. Their existence expands our understanding of where life is possible, suggesting that the habitable zone extends far beyond what we once imagined. Some microbes live in frozen Antarctic ice for thousands of years. Trapped in glacial ice, microscopic organisms can enter a state of suspended animation that persists for millennia. When scientists drill ice cores from Antarctica and melt samples from layers thousands of years old, they sometimes find bacteria that resume growing when given warmth and nutrients, awakening from a sleep that began before human civilization. These organisms survive by allowing their metabolism to slow to almost nothing, waiting with patience beyond human comprehension for conditions to improve. They are living time capsules, proof that life can pause itself and wait through ages of darkness. Microscopic bacteria thrive in the deepest ocean trenches. At the bottom of the Mariana Trench, where the pressure is more than 1,000 times greater than at sea level and sunlight has never penetrated, bacteria live in the sediment and water, adapted to conditions of crushing force and eternal darkness. They feed on the rain of organic matter that drifts down from the surface and on chemicals seeping from the seafloor. Their existence demonstrates that life has colonized every available habitat on Earth, from the highest peaks to the deepest valleys, exploiting every possible source of energy and adapting to every extreme. Microbes have been found living kilometers beneath the Earth's surface. Drilled deep into the rock beneath continents and oceans, scientists have discovered ecosystems existing in darkness, nourished not by sunlight, but by chemical reactions between water and minerals. These subsurface organisms grow incredibly slowly, dividing perhaps once per century, living in a realm where time moves at geological pace. They may constitute a significant portion of Earth's total biomass, an entire hidden biosphere beneath our feet, unknown and unsuspected until recent decades. Some bacteria can survive intense radiation that would kill humans instantly. Deinococcus radiodurans, nicknamed Conan the bacterium, can withstand radiation doses thousands of times greater than what would be lethal to humans. When its DNA is shattered by radiation, the bacterium simply reassembles the pieces, reconstructing its genome from fragments with remarkable accuracy. This extraordinary resilience may have evolved as a side effect of adaptations to extreme desiccation, but it has made these bacteria candidates for bioremediation of radioactive waste, sites and potential travelers to other worlds. Microscopic organisms have existed on Earth for over 3.5 billion years, long before plants, animals, or fungi appeared. Microorganisms ruled the planet. For billions of years, Earth was a microbial world, its surface covered with bacterial mats and its oceans filled with single-celled life. These ancient organisms transformed the planet, creating the oxygen atmosphere that would later allow complex life to evolve, cycling nutrients, building the first ecosystems. We are latecomers to a world shaped by the microscopic, inheritors of a planet engineered by beings we can barely see. The oldest fossils are of microscopic bacteria. When scientists examine rocks from the earliest epochs of Earth's history, the only traces of life they find are microscopic, fossilized stromatolites built by bacterial communities, chemical signatures of metabolic processes, and occasionally the preserved remains of individual bacterial cells. These fossils push the origin of life back to within a few 100 million years of Earth's formation, suggesting that life arose almost as soon as conditions permitted, and that the microscopic world is not a simplified version of life, but its original and most enduring form. Microscopic cyanobacteria created Earth's oxygen atmosphere. The air we breathe exists because of microscopic organisms. Around 2.4 billion years ago, cyanobacteria evolved the ability to perform oxygenic photosynthesis, splitting water molecules and releasing oxygen as waste. Over hundreds of millions of years, this oxygen accumulated in the atmosphere, transforming Earth from a world of methane and carbon dioxide into one where oxygen-breathing organisms could evolve. Every breath we take is made possible by this ancient innovation, by microscopic cells that change the composition of an entire planet. All visible life descended from microscopic ancestors. Every tree, every animal, every fungus, every plant shares a common origin in single-celled organisms that lived billions of years ago. The complexity we see in the visible world emerged gradually from microscopic simplicity through countless generations of mutation and selection. Humans and bacteria are cousins, separated by billions of years of divergent evolution, but sharing the same fundamental molecular machinery, the same genetic code, the same basic chemistry of life. We are the descendants of the microscopic, complexity built upon a foundation of single cells. Your cells contain microscopic structures that were once independent organisms. Mitochondria, the powerhouses of your cells, were once free living bacteria that took up residence inside other cells billions of years ago. This ancient merger, called endosymbiosis, transformed both partners into something new, creating the complex cells that would eventually give rise to all animals, plants, and fungi. The evidence for this origin is written in the mitochondria themselves, in their double membranes, their bacterial-like DNA, their independent reproduction within cells. We carry within us the legacy of ancient partnerships, microscopic collaborations that became permanent. Every atom in your body has existed for billions of years. The carbon in your muscles, the calcium in your bones, the iron in your blood, all of these atoms were forged in stars that lived and died before our sun was born. They have cycled through countless organisms, been part of ancient seas and primordial atmospheres, existed in forms beyond imagining. At the atomic level, you are made of stardust, ancient beyond measure, assembled temporarily into the pattern that is you, before dispersing again into the world. The atoms remember nothing, yet they contain within their structure the history of the universe itself. The water molecules in your body have traveled through countless organisms. The water you drink today may have once been part of a dinosaur, a tree in an ancient forest, a cloud over a vanished ocean. Water cycles endlessly through Earth's systems, evaporating and condensing, flowing through rivers and rains, absorbed by roots and exhaled by leaves. Each molecule has made this journey countless times, passing through the bodies of innumerable living things, connecting all life across time through this shared substance. You are not separate from the world, but part of its circulation. Water flowing through you as it has flowed through everything. Microscopic carbon atoms in your body may have once been part of a star. Carbon is created in the nuclear furnaces of massive stars and scattered into space when those stars explode as supernovae. The carbon atoms that form the backbone of every protein, every DNA molecule, every cell membrane in your body were born in stellar fire billions of years ago, drifted through space as cosmic dust, and eventually coalesced into the matter that formed our solar system. At the microscopic level, you are made of ancient explosions, stellar remnants given temporary form, a pattern of organization that borrowed atoms from the cosmos and will one day return them. At the quantum level, particles exist in multiple states simultaneously. When we descend to the scale of atoms and subatomic particles, the rules of everyday reality no longer apply. A particle can exist in a superposition of states, simultaneously here and there, spinning both directions at once, until the moment it is observed and forced to choose. This is not a limitation of our knowledge, but a fundamental property of reality at the microscopic scale, a strangeness confirmed by countless experiments. The solid, predictable world we experience emerges from this quantum uncertainty, order arising from a foundation of probability. Microscopic particles can be entangled across vast distances. When two particles interact in certain ways, they become connected in a manner that transcends space, their states linked even if separated by light years. Measuring one particle instantly affects the other. A correlation that seems to violate our intuition about how the universe works, yet has been verified experimentally again and again. This quantum entanglement operates at the microscopic level, but reveals something profound about the nature of reality, suggesting that separation is not as absolute as it appears. That connection can persist across any distance. The boundary between the microscopic and the visible is itself invisible. There is no sharp line where the microscopic world ends and the visible begins. No moment when enough microscopic things gathered together suddenly become visible. The transition is gradual, a continuous spectrum from atoms to molecules to cells to tissues to organisms. The categories we create, microscopic, macroscopic, visible, invisible, are conveniences, useful fictions that help us organize our understanding, but nature recognizes no such divisions. The world is one continuous whole, and the boundaries we perceive are artifacts of our limited senses. And so, our quiet journey through the microscopic world draws gently to a close. We've drifted from the invisible dance of atoms and molecules to the resilient water bears walking through moss, to the glowing tides sparked by dinoflagellates, to the vast colonies of bacteria that make life possible. This hidden universe has been here all along, closer than breath, smaller than sight, woven into every moment of your existence. It can be humbling to remember that you are never truly alone. Right now, trillions of microscopic organisms are living peacefully within you and around you, carrying out their ancient work. The oxygen you breathe was made by microscopic life. The soil beneath sleeping forests is alive with invisible threads connecting tree to tree. Even the atoms that form your body were once scattered across the cosmos, now gathered here, now part of you, vibrating with the same energy that moves through all things, seen and unseen. But for now, there is nothing more to do. Just allow your body to sink deeper into rest. Let the day fall away behind you and let the calm widen within you like the infinite smallness of space between atoms. There is no need to hold on to anything. The microscopic world will continue its work while you sleep, quietly, perfectly, endlessly. If you've enjoyed it, you can gently like, subscribe, or leave a thought in the comments. It helps others find their way here too. Sleep well and dream among the invisible wonders. Good night.